George and Eric to come up on stage and join me in a second, and then we're going to have the welcome message from Governor Gavin Newsom. So first I'm going to introduce George. You know George Church because George Church 2 years ago did the fireside chat with Vinod Khosla, and a year ago George Church did the fireside chat with Steve Wozniak. George is professor of genetics at Harvard Medical School and Massachusetts Institute of Technology. He is widely recognized for his innovative contributions to genomic science and his many pioneering contributions to chemistry and biomedicine. He developed the first direct genomic sequencing method and helped initiate the Human Genome Project, leading to a revolution in science and medicine whose impact is still being realized. Please welcome to the stage, George Church. Welcome back. And joining George today, Eric Schmidt is one of the world's most successful business leaders. As CEO of Google from 2001 to 2011, he is arguably one of the internet's most important people. Eric is well-known philanthropist, particularly in the areas of science and natural resources. In 2010, he co-founded Innovation Endeavors, an early-stage venture capital firm that believes that recent advances in science and technology will create exponential rates of change and emergent ecosystems in a new world. Please welcome to the SynBioBeta stage, Eric Schmidt.
Thank you. Thank you.
And if I could have my iPad, we're going to be doing audience Q&A. So if you have questions, go to synbiobeta.com/questions. And towards the end of the fireside chat, I will be flipping through and picking some of the best questions that we're going to be asking. But before we do that, great, thank you. But before we do that, we have a very special message from the California Governor, Gavin Newsom. And if you've been reading some of the data that we've been putting out in the Forbes column lately about the growth of the American bioeconomy, the growth of the global bioeconomy, but also the growth of the California bioeconomy, we showed that there was over $10 billion invested in the synthetic biology industry over the last 10 years. And very surprisingly to me, $5 billion of it was invested in California, and $4.5 billion of it was invested in Massachusetts. And the next closest state was, I think, $350 million, which was North Carolina. So a huge disparity in where this investment is taking place. So if we could cue up the video. We sent the data to the governor's office to look at this, and we also put up what we called the Bay Area Ring of Fire. This about 150 synthetic biology companies all around the Bay Area. And so in response to that, we got this fantastic welcome message. So let's play the video.
Hey everybody, Governor Gavin Newsom, former mayor of San Francisco. Welcome to San Francisco. Welcome to the state of California for the SynBioBeta Annual Global Conference. Thank you for being at a city and being in a state that prides itself on being on the leading and cutting edge. We love to say about San Francisco and California, the future, it happens here first. We are America and the world's coming attraction. So it's appropriate that you all descended upon San Francisco. It's also appropriate for another reason. We're the birthplace of biotech. We've been on the leading and cutting edge of biotherapeutics, bioinnovation, at least the inspiration, but we haven't necessarily seen the manufacturing. And that's why I'm here on this video, to make the case not just for San Francisco, but the entire state of California, particularly as it relates to synthetic biology, biotherapeutics, bioinnovations, but engineering biology that brings you all together. So thank you for your innovative and entrepreneurial spirit. Thank you for being here today. Thanks for enlivening the senses and imagining a future that's brighter, healthier, and more environmentally sustainable. Have a great conference, everybody.
Fantastic. So thinking about the political landscape that we live in right now, I just became a US citizen about 10 days ago.
So congratulations.
Thank you. And I'm still a British citizen as well. But I know where I'm putting my eggs right now, unfortunately. Why aren't there more scientists engaged in politics?
George, is that your next?
No, definitely not. But they're more than ever before. There was this March for Science that mobilized scientists, and it was I participated in it, and it was the weirdest feeling to give a talk to 50,000 people who would interrupt with applause in the middle of what you were saying. You know, it's like this. Politicians take this for granted, but scientists, you're supposed to wait until the end. And anyway.
I don't think we collectively understand how powerful the decisions that were made by Vannevar Bush and others 50, 60, 70 years ago that set what we're doing today. Pretty much everything that you experience today of modernity came through some form of government-funded research or innovation and so forth. And the American model is this unique model, which you see here at the conference, of super smart people, super smart universities, and innovation. And the federal government has played an outsized role in that. I can say, for example, I was supported by NSF grants as a graduate student. I was supported by DARPA grants as a graduate student because, frankly, I needed the money, right? I'm very grateful to that, right? So were the projects that I was on. And today, the same thing is occurring. The difference is that in the area of health, for example, the amount of money that we spend collectively as a country on health has gone down roughly 30% in adjusted dollars in the last 5 to 7 years. Now that's insane because the number of people who are getting older has increased as a percentage. The president's current budget reduces science by something like 5%. The original proposals were worse and people worked hard to get that. So you ignore the actions of politicians and government as a scientist at your own peril in the form of lack of grants, right? These discoveries take funds. Private philanthropy can make up some of it, but not as much as the government contributed.
Can you talk a little bit about some of the work that your philanthropic organization is doing, in particular the Schmidt Futures Initiative?
Well, in our case, not to go into too much detail, you can read about it. I've gotten very interested in AI-enabled science, and the simple argument is you have these brilliant scientists who are not computer scientists, and they need computing power, and they need computer scientists to help them engineer these systems, because all interesting problems in science seem to have an awful lot of data and an awful lot of confusion, right? It's no longer, you get your microscope and you look very precisely and you see something and you go, holy, amazing, and you get your Nobel Prize. It's much more complicated now. They're algorithmic approaches. The other thing I've gotten very interested in is global talent. One of the other errors that's going on in the country is people are not understanding how powerful global talent is to the American innovation engine. If you look at the graduate students in top universities, a very significant number of the graduate students are foreign-born, and they make huge contributions in science. And so globally searching for that talent, globally identifying it, globally getting them to the US is another goal.
George, can you talk about the makeup of your lab and the role of immigration?
Oh, it's extremely diverse. It is dependent upon that globe. We have quite a number of Iranian and Chinese postdocs, for example. And I think it's vital. I mean, it helps you think out of the box. It helps you tap into the best and the brightest worldwide. So we certainly no longer have a monopoly.
And it's easy to make the argument. Would you prefer to have these top people working for George and then going off and creating a company which creates huge wealth in America and pays lots of taxes, or would you prefer them to be somewhere else and do it in another country? Your choice.
I wish it was just their choice, yes.
George, can you name a couple of those companies of foreign-born graduate students who've come in and then ended up?
Probably one of the better examples is Luhan Yang, who was a graduate student and a postdoc with me. She was almost thrown out of graduate school because of her English skills.
The best people are, not for English, but.
and also, it was confused with lack of understanding. Anyway, she formed eGenesis in Cambridge and Kihan in Hangzhou, China, and she's CSO for one and CEO for the other, and commutes back and forth. Anyway, she's a force of nature that's amazing. These companies are making organs that are already in preclinical non-human primate trials, and they're very, very successful, but it involves synthetic biology where you're making dozens of edits to the genome in a way that you have to have very healthy pigs at the end of the day.
And Luhan has spoken at our previous events about this. Eric, can you talk a little bit about the Deep Life initiative that you recently launched with Innovation Endeavors?
Well,
and maybe just give people a broad reminder what Innovation Endeavors is.
Well, again, I'm associated with a venture initiative around synthetic biology and so forth, which we call Deep Life. And the core idea here is that all of these initiatives will be accelerated by computing and computer science approaches of one kind or another. And one way to understand synthetic biology is it's the marriage of biology and computing, and you need both. And what's interesting is I had always thought that it was pretty straightforward. You would take everything in the soup, you'd sort of lay it out as a choice, you would run computer algorithms over it, and then the answer would become apparent. And this, as you know, is laughable to you. It's much, much more complicated. And so we're trying to fund companies that are in the multi-stage of using both. They always have a combination of some kind of biological testing regimen where they're actually doing something in a lab, and then they have either direct or indirect things which they're measuring using machine learning to come to some test. And the key idea is the faster you can do the test cycle, the faster you can iterate. Now, one of the complaints I have about biology is that you can't iterate digitally, because we don't have, and I'm looking to you now, you don't have perfect digital simulations of biology. So you're still stuck with the try it, test it, do an assay cycle, right? And you've gotten better at doing it quickly. But the faster you do that, right, then you get into the digital world and you get huge gains very quickly. The reason my industry has done so well is we operate in an all-digital world. So it's easy, for example, to come up with excellent, movie recommendations or YouTube recommendations or what have you, because we have millions of data points of people like you that allow us to train to make a good suggestion for you. We don't have the analogous amount of data in biology yet.
George, is this something you're working on?
Sort of, yeah. So, I mean, where you can tighten up the speed at which you're doing cycles by eliminating cellular and organismal generation time, doing things in vitro. You can get high parallelism, so when in a sense the biological system is a computer of its sort of natural computing, where you can put in trillions of very complex protein folding problems that solve the solution and select out and then use Darwinian selection to pull out answers. So you're doing something that seems a lot faster than, doesn't seem quite as limited as it may, as digital might be.
Well, so you're making an argument that eventually your world should be able to compute faster,
right?
Because you can
for certain problems.
For certain problems, you can use the nature of the quantum world and parallelism in biology to actually compute faster.
Exactly.
And the sooner that happens, the quicker I'll retire. Right.
Yes.
George, what do you think computers can teach us about how to program biology? And what do you think biology can teach us about how to program computers?
Well, I mean, biology is often used more metaphorically than in detailed mechanisms, but it's the basis of neural nets, the basis of machine learning, but again, metaphorically. But there are efforts, like IARPA launched an effort to understand a cubic millimeter of visual cortex in order to make that much more literal inspiration for algorithms. So IARPA doesn't care much about the biology, it cares about the algorithms that came out of that $100 million project. The other way, I mean, it's totally obvious that every day we use, computational biology, statistical methods, machine learning methods to analyze very, very growing data sets, imaging data sets, genomic data sets, synthetic biology data sets.
Eric, you talked about the applying principles of engineering to biological systems, going through the design, build, test cycle. We have a workshop tomorrow that Berkeley Lights, one of the digital molecular biology companies, is putting on to try to bring together the industry to talk about benchmarking, so that they can look at the design, build, test cycle of different strain engineering processes and be able to compare them. Can you give us some historical perspectives of the waves of innovation that you've seen coming through Silicon Valley, and particularly efforts around benchmarking, where the industries got together and rolled up their sleeves and said, hey, this is kind of pre-competitive, or it's competitive, or we're going to put together research dollars into this?
I think our examples are not as complicated as yours for regulatory reasons and so forth, but I think they're illustrative. The creation of the internet itself, which is well known, was an act of goodness from a whole bunch of players. And I was always struck by, in the first version of the internet 30 years ago, that companies were perfectly willing to accept packets to route without asking for a charge. In other words, the people who were doing this design must not have told their CEOs they were doing this. They must literally have said, I'm just going to sneak this thing in, and we're going to sort of do hot potato routing where we're going to make it all work. Today, people understand this, but it was sort of a pleasant surprise. And the fact that the network held under enormous sort of competitive pressure is an example of that kind of collaboration. In the internet, there was a group called the IETF, which did similar standards. In most cases, standards in my industry were done by simple examples. So what would happen is somebody would implement an open source implementation of something, and that open source implementation, which would be free to everybody, would be used. More recently, there was a group called ImageNet that was formed roughly in 2011. They created an image database and ultimately led to a set of contests, and those contests were fundamentally about image recognition against this database of images. And again, it was done in the open, done globally, and so forth. I wonder if the same, and George is familiar with a number of examples of this in his field, where if you could get that at scale, you could accelerate the communal learning of it. If I look at it, biology, you don't have enough data standards. The data that is collected is largely proprietary. It's in corporations of one kind or another. You don't share your failure information, you just share your success information. So there's a whole bunch of things that if it were a collective university, which I understand it's not, that would occur more quickly. So collectively, biology could agree to interoperable standards, faster iteration, that would help everybody. And you could still keep your proprietary advantage. The biggest problem that I always had was in explaining how do you make money under open source? Well, if you give it away, you can't possibly make money, and yet it's obviously not true. It took a very long time for people to understand that this sharing model of software ultimately produced proprietary businesses, which it has.
George, any thoughts on a proprietary open source layer for biology?
Yeah, there are all kinds of excuses that are given for being different from the internet. We started Personal Genome Project in 2005, in part anticipating and responding to the claim that you could not make human biomedical data publicly available the way you make Wikipedia available for all kinds of reasons, and showed that it does work in multiple countries now. Many of the other data sets have been forced to be open by grant and journal practices. For example, accession numbers were required sort of as I move through fields, first in crystallography, then in genomics, and then in transcription data. So those are fairly large data sets at this point.
And there's another one called the Human Cell Atlas, which is on its way. The biggest violators, by the way, are not in genetics and biology, but in the medical field, where it wasn't until recently where they had common numeric codes for the same data, right? So again, it's very hard for computers to make progress in medicine until you have common data formats.
So I want to talk about AI and synthetic biology, but before we do, I want to talk about strong AI versus weak AI. And whenever I have an important fireside chat like this to prepare for, I often call a friend. And this time I called Rob Reinhardt, the CEO of Soylent. I said, give me some clever questions to ask George and Eric. And he said, well, ask them about strong AI versus weak AI, and then ask them to explain to the audience what this means. And I have no clue what strong AI or weak AI means, so you can explain to me as well.
So there are different ways of describing the distinction. I don't personally like strong versus weak, because the weak stuff is what's powering everything around us. So generally, the weak term refers to AI that is designed to solve a specific problem. So translation, image recognition, those sorts of things. They're narrowly defined. Another term is narrow AI. And strong AI is the conception that the AI will be able to either be self-directed, self-learning, self-determinative. And there are very few examples of true strong AI today. There's a raging debate in my industry as to when this will occur. One of my friends, and I said, I want to know what is the month and what is the year? And he said October 2044, which is conveniently 25 years from now, which is obviously a random number. And he said that is the median of the dispersion of everyone's answer to when will what is known as AGI arrive. So the important point is it's not in the next month.
Right.
Is a conventional computer capable of strong AI? Is a biological computer capable of strong AI?
So one of the open questions is how much computation is going to be needed for general intelligence. We know that there's increasing returns to using more computers for narrow AI, and we also know that there's increasing returns for the use of these AI accelerators, which are all the rage. Google, for example, has some, their competitors have them, and so forth. And what they do is they essentially take the specific algorithm, which is known as gradient descent, and they make it run much, much faster. Eventually, those things will be affected by Moore's law. Right? Eventually, there are limits to that. And it's an open question what happens after that. There are many people who think that quantum computing, because quantum computing can do gradient descent really well, will be able to solve the problem. Perhaps there'll be biological mechanisms to do this, or perhaps it will be a hard problem for a while. I don't think we know yet.
George, any thoughts?
Well, we certainly have a proof of concept experiment that you can make a biological computer that does general intelligence, which is inside these crania. I think part of the reason that we get excited about computers is not necessarily just because they're faster, because they also sometimes consume extra energy to achieve that speed, but because we can do experiments on computers that would be unethical to do on humans. But as they get closer to general intelligence, then we'll probably have the same set of constraints. And probably there will be growing interest in messing around with human cognition, not through the germline, but to deal with cognitive disorders. And then that will have some kind of side benefits to everybody.
Can I add that I think that while this is a super sexy question, the reality of what's going to happen is much more into an assistant model. I was talking to my friend who's a physicist, and I was telling him that we're going to have this incredibly smart system, and it'll be able to do physics, and won't you be happy? And he says, I don't need that. What I need is a computer that can read all the physics papers and then suggest things for me. And I said, why? He said, because you can't imagine the amount of stuff that people are generating, and nobody can keep up with it. And I think that's much more illustrative of what will really happen. So that the next generation of fantastic people, and by the way, I should say, George, every single person who is brilliant in this field has gone through your lab. That's how profound your contribution has been. So if we go back to, if we go back to this 10 years from now, the likelihood is such a person will have an assistant. They will help read the information, will help them look at scenarios. It's a long time before that person gets replaced. But in the intermediate time, long time meaning many, many decades if ever, but in the meantime, computers will make it much, much easier for people to deal with the incredible complexity that we're generating.
So I'm going to take a few questions from the audience, and there's a lot about politics. I think we touched a nerve there, but I'm not going to go back to politics. Maybe we'll do it later. What are you most excited about in synthetic biology, and what are you most worried about? George, do you want to take that?
Well, I worry about everything, even in certain sense that the things that are more exciting are also more troubling. But we have a pretty good track record of persisting in spite of all the negative things that could be done, because we just want to we encourage, especially in the synthetic biology community. I think we've done a good job through iGEM and other human practices, SynBioBeta, of encouraging a positive way rather than having the equivalent of robot wars. So I'm most excited about things having to do with organs, including brain organoids, from either by humanizing pigs or by synthesizing them in the lab. And there's this is a new Moore's Law that's going exponential.
Can you give some specific examples of how fast that's moving?
Well, for example, we just editing of organs and organoids. We were stuck at 2 edits, 2 genes at a time. A couple years later, we were doing 62. A couple years after that, we're now doing 26,000 of them. That's edits. We also gone from organoids as being kind of like, well, they look kind of cute at half a millimeter, but they get necrotic in the middle, to now we're making things that represent late onset Alzheimer's, schizophrenia, bipolar, in ways where the patients and the age-match controls are dramatically different. Some of these things we haven't even formulated the Moore's Law curve for it, but you can feel it just as real as the 10 million fold that we brought down sequencing and synthesis costs.
Eric?
I think I'm most interested in the aspects of generative design that now apply to synthetic biology. So what I thought was going to happen was, you'd have this deep learning model, you'd have all this data, you would crunch it, and you'd get insights. But the data is so messy, it's so imprecise, it's so non-computable in my terms, that the latest approaches seem to be to take the real data and then do something which is called a GAN, where what you do is you basically try to build a digital version of the data you have, and you have something which generates candidates, and you have something which compares to reality, and eventually you get something which is digitally similar to the things that you have. And that digitally similar thing is of a uniform structure, uniform data, and has the essence of the data that you have. So this technique seems to be at the basis of most of the synthetic bio algorithms that I've seen. And I'm sure there'll be more. But people have sort of just discovered this in the last year.
I want to talk a little bit about the company that's doing the recoding. Is it 64?
64 minus X.
64 minus X. George, can you explain why it's called 64 minus X?
Right. So Lexi Rubner is, I think, here somewhere.
I saw her earlier.
CEO. And 64 is the number of codons, so 4 to the 3rd power. And it is ACGT in every possible combination of 3. And it helps us go from the DNA RNA world to the protein world, where the 20 amino acids are in this redundant code. Because it's redundant, you can swap any one of the 64 codons in principle for any of the other ones, and you can move them in mass from one codon to another. And then once you do that, you can now rip out the machinery that used to service that codon or pair of codons, which before would have been lethal. Now you can rip it out, and when you do that, you get 3 really cool new behaviors. You're not just synthesizing a genome for kicks. You're doing it. You get non-standard amino acids with a whole lot of new chemistry, new fluorescence, new redox, and so forth. You're getting biocontainment. We've shown biocontainment to at least 16 orders of magnitude. And then the coolest, I think, is that you can make any organism resistant to all viruses simultaneously, all naturally occurring viruses, including ones you've never seen before. So if another Ebola crawls out of the jungle, you are already ready for it in principle. So that's 64-X is to pursue some of those opportunities.
And another company that's doing optimizing DNA code is Grow Bioscience.
Correct.
Tell us about Grow.
So Grow is a little bit ahead on that, but they're taking the first of those three applications, which is non-standard amino acids, and developing new use cases for them. So we can now make a non-standard amino acid extremely efficient, about as efficient as a normal amino acid. And so you can make things like disulfide bonds, which are very messy in biotechnology because they get broken very easily or cause misfolding. We make diselenide bonds. So selenium is one rung down on the periodic table from sulfur, and these are strong enough that they persist in the cytoplasm, which normally melts the disulfide bonds. That's just one example, and that can be used for making drugs like DNA-1. It's important for cystic fibrosis, a whole variety of biotechnology, antibodies, and so forth, which depend on disulfides.
So if we think about both of those companies in some way optimizing the biological code, Eric, can you compare and contrast that to the time that's spent as a software engineer writing code versus optimizing existing code? Maybe think about that in the biological context.
I'm not sure they're directly related. My reaction to George's report is, I just want all of that sooner, right? And I'm sure everybody feels the same way.
Yeah.
And if you look at the startups in SynBio, it just takes longer. It takes more capital and takes longer. And it just seems like if we could collectively shorten that time, either with more money, better algorithms, better testing programs, or better sharing, that would be good for everybody. In my world, almost all the code is rewritten constantly. And one of my great personal frustrations is that in every case, the code that is interesting was written by 2 or 3 people, and now it takes 1,000 people to do the same thing. And for the life of me, I don't understand why in my career, it's always been the case that 2 or 3 people can somehow get the first version, and by the 10th version, it's 1,000 people. And if you look, this is true across the entire industry, across every platform.
Wow.
And I can understand 100, but why 1,000? And to me, that's an example of a failure of abstraction. In other words, there must be something that we're not doing that then allows people to become even more productive than they are. And I don't know if that's analogous to the way biological research is going, or whether there is also possible stepping stones. But we're very good at adding complexity. We're not as good at building the ladder above the complexity to the next layer. And proper computer science is that whatever the muck was here, you build another layer that unifies it. And then when that gets to mess, you build another layer. And the faster you move up, the greater the power of the system.
Now, could it be...
I'll just add a little frivolous biology here. It's kind of like a new mutation. It's the next step in evolution. Requires one individual.
Exactly.
And then to make it actually work so that it's suitable for fine tuning and so forth takes tens of thousands of individuals. Right. So there is a precedent.
Now, could it be that nobody's funding research on that problem, that once these programs get so complex?
This has been worked on for my whole career as a computer scientist, and I don't think we have the breakthrough yet. And one answer would be to have the computers write the code. So many people believe that this next generation of computers, you'll essentially write a description of what you want, which is fairly specific. It's not just random, and that it'll be able to generate enough code to really fill it out. There are many examples of this already, which takes, for example, Python and turn it into higher performance code and things like this. And you could imagine that will help solve my problem. But at the moment, let me just observe that there are thousands and thousands and thousands of programmers rewriting the technology and ideas that really came from 2 or 3 people. We've got to come up with a good solution for that.
The argument for 20 years of synthetic biology has been that biology is difficult to program because we've never focused on the tools and technologies and platforms and abstraction layers to make it easy to engineer. Do you agree with that, George?
Oh, that's partly it. I mean, I think synthetic biotechnology has gone very fast because we've been riding on the coattails or drafting behind the electronics and computer industry. We inherited computer-aided design and computers themselves and so forth. So we actually have gone, that's the reason we've gone faster than Moore's Law for at least reading and writing DNA and things related to that. But you know, I think that biology is capable of going quite quickly.
But there's another possible answer, which is that the source of innovation in biology, at least for a while, has been the smart faculty member and the incredibly smart graduate students and the discovery in the lab working very, very hard. And to the degree that we can systematize that, it is helpful. So for example, there's something called a Jupyter Notebook now, which people use to record all their results, which allows people to test things. In protein folding, there's a language or a system called Rosetta. I mean, there's a series of platforms that look more and more like the kind of platforms that we take for granted.
So what are some of the opportunities for step functional improvements that you think we could imagine over the next 15 years in our ability to engineer biology?
Well, combining this with your previous question, I think one of the things that makes biology hard is not just the lack of engineering principles, which I think synthetic biology is trying to address, but it's that we inherited some very sophisticated technology. In the end, technology in metaphoric quotes. In the end, part of the reason it takes 10,000 people is there's a lot of corner cases and a lot of fine-tuning. In biology, a lot of that's already happened. It's given to us as a gift. We don't have to predict CRISPR. It's given to us. We have to just understand what we can do with it. And this happens again and again, DNA polymerase, ribosomes, just all these things that would take, I think, millions of human beings to figure out from first principles. Something equivalent to engineering. So you can think engineering is about theory but it's actually also about trial and error. And evolution is about trial and error. So we get these incredible gifts from nature that we're just beginning to understand, and to some extent ape and mimic with our own efforts at accelerated evolution and accelerated developmental biology.
Eric, do you agree?
I do. Biology is different because there is a given case to start with, right? That you can study and study and study. I was struck by both how much we don't understand about how biology works and how much progress has been made, especially in the last decade, given how little we actually understand about the underlying mechanisms. And I don't know whether it's a good thing or a bad thing, but I think eventually we will fully understand it. Right? It will eventually be fully understood.
And it's amazing how much we can do before we have full understanding. Right. Like my favorite example is smallpox vaccine. We didn't understand virology or immunology at all, and we nevertheless happened to get that to work. I don't recommend it in general, but it does happen.
So I asked a question earlier about what are you excited about for synthetic biology, and it clearly wasn't, the answers clearly weren't wild enough, because I've got a question from Ryan Shea in the audience who says, what's the most wild, impactful, and underappreciated application you're excited about within the field of synthetic biology? I think they want you to think really far
I'm just
in the
waiting. George talked a long time ago about DNA being used as disk drives, and I'm really looking for disk drives that are about 1 million times better than the current ones. So I'll start with that. Right.
And I'll respond to that before I go on to my wild and crazy thing. I think the thing about new technologies is that they're often tried to pigeonhole to fit into previous problems and technologies, and the ability to store data in DNA form, which gives us longevity, gives us compactness, low energy, and interfaceability with biology, isn't necessarily to compete with disk drives. It's not even necessarily, it is digital, which is a nice start. It's intrinsically digital. But one of the things we kind of backed into was storing all the information of the developmental biology of a mouse from egg to adult, about a terabyte of information in 30 nanograms of DNA. So about 1 billionth of the mass of the mouse is dedicated to storing this developmental energy. It's a very specialized case but it's not a toy case. It's actually interesting, and we learn things about developmental biology. But I think that's where we're going, is a physician will have this black box of the patient, and they can go in and they can get a record of what went wrong, for example.
And can I just add to the scenario, in terms of things that to be excited about from my perspective, in biology there's an awful lot of people who are trying to understand how human systems in the brain and so forth work that will help inform us algorithmically. So I don't think deep learning and neural networks is the end of the analysis of the human brain. I think there's much more complicated things going on. And the studies that people are doing, whether it's a closed system around the eye, and the way the vision works and so forth, will undoubtedly generate algorithms which are very powerful that we don't fully understand right now. Go ahead, I interrupted you, George.
No, I was basically done with that.
Well, I think we wanted the big, crazy, wild idea from you.
Crazy, wild idea.
You heard it here first at SynBioBeta, right?
Yeah. Well, this is not necessarily, this is probably 1/4 baked rather than half baked, but I was very interested in developing, getting us off the planet, getting backup for us, because yay, it's okay. And, we need a backup. And even if we're not destroying ourselves, we are at risk for solar flares and supervolcanoes and asteroids, which are not our fault. And so one of this is making colonies on Earth to prepare ourselves for going to Mars, rather than setting up, because on Mars, if we make a mistake, it's a disaster. But but we want to do thousands of colonies on Earth, small ones, to see what works and what doesn't work in terms of closed systems, total recycling, psychological, and so forth. So that's one thing. And then to go even further, I'd like to get to interstellar, and that means we're probably, if we want to get feedback on that, we're probably going at a fraction of the speed of light. And there have been proposals of getting our electronic probes down to 1 gram, breakthrough star shot. And I think that's not far enough. Getting them from kilogram to gram scale, I say get them down to nanogram scale. And the nanogram is actually pretty big in biological terms. It's an egg or a small multicellular cluster. Make it radiation resistant, send it out there in such a way that solar wind and gravitational fields will bring it into a planet. And then, you can send out billions of them for the price of 1 gram. And then they have to build telecommunications, like a bird builds nests. You teach this nanogram how to build something to talk to us. So maybe that's 1/8 baked. Yeah.
Meanwhile, back on Earth, can I just ask you guys to build bacteria that consume CO2 and don't produce output, please?
Yeah, that's true. There are some. Cyanobacteria do a good job of that. We just need to harness them.
Yeah, we need them at scale, and we need them in the next 10 to 15 years.
If you were to tell your younger self one profound lesson that you've learned along your journey that would have helped you the most, what comes to mind? Who wants to go first?
Well, my answer is that I was lucky, and that I was very early in computer science before computer science existed, and I rode that wave. So my advice to myself would be to be lucky and look around, not unreasonable, and look around. I'm convinced that synthetic biology is such a luck path because it's at its infancy. You can still get together in a conference. You can still talk to each other. People tend to know each other. There's not that many players. And you can build huge companies and huge careers over your lifetime if you start then.
Fantastic. George?
I think I must have gone back and told my younger self something, because I also felt fairly lucky. But I think I lacked confidence. That may not seem apparent at this particular moment, but there were many times where things did not seem well aligned for what I was doing. It seemed like it was not well thought out. So maybe I would go back and say have more confidence, but it probably would have resulted in arrogance or something. And so it's probably not a good idea to go back and talk to your former self.
But one of the ways there's lots of discussion about how to create luck. And one way to create luck is to be in a community of people who are stimulating. Right? So if you look at the structure of biology and genetics and so forth, they're largely centered around the greater Boston and the greater San Francisco area for a reason. As much as we have telecommunications and distance learning and so forth and so on, there's real benefits to being surrounded by it. It builds a community and so forth. Lots of people have studied why the innovation ecosystem in California works so well, and it's a lot because of proximity. Right? So one answer, if you want to increase your luck, is move to an area where there's lots of lucky people.
Right. True.
So that's a good segue to a question from Zal. I presume this is Zal Bilimoria at Refactor Capital. And Zal says, do either or both of you believe that massive biotech startups can be built outside of the Bay Area or Boston?
Well, yes. I think that BGI is a fairly good example of both sequencing and synthesis. It's pretty far away from California and Boston in Shenzhen.
No, but look at San Diego, right, as an example. I think that there's a set of components that you need, and good weather is not one of them.
Yes, it is. Says the British guy.
Last time I checked, you were at Harvard.
Yeah.
It's clear that you need a strong university. It's clear that you need some form of startup culture. You need a country where the laws allowed for limited investments. Many countries aspire to have innovation ecosystems. When you actually look at their laws criminalize failure or don't allow for limited partner investment and so forth. And I'll talk to the leader of the country and they'll say, well, we're going to create a Silicon Valley of this. And I said, no, you're not, because your laws don't mirror the state of the art. And so there's a set of things. It's not just one thing. It's proximity, it's near university, access to capital, the right kind of people, the right kind of culture. There's a lot of people who claim that the original computer culture here was stimulated by the free speech movement, right? And that the sort of anti-authoritarianism of the 1960s was part of what created the startups that ultimately became Apple, Sun, and so forth.
Ali Nouri from the Federation of American Scientists asks, the US government is cracking down on foreign-born scientists alleged to have transferred research data back home. Are we overacting, or is there a security concern here?
So you may have, we probably agree on this. Let's see if we do. There was a bargain made a long time ago. And the bargain was that foreign students would work in our universities. Most of them would stay in the United States. Most of the benefits would come to the United States. Some of them would go back. Some of the ideas would go back. That in general, it's better for America to compete and lead with that model. That's roughly the bargain that was made, whether it was explicit or not, and it's worked really well for us. There clearly are cases of espionage which are illegal and they should be prosecuted. But I am strongly opposed to labeling people who are here under that bargain, who are working hard as part of their vision of the American dream as foreign-born, to somehow being discriminated against.
Great answer.
Yeah, and I agree. And it's a fairly exceptional case. Most of the things that we do are perfectly capable of being open sourced. And so all they're not taking secrets, they're just taking know-how that they achieved in the process of creating this open source and know-how that remains in the laboratory. So yeah. I think you take a few strange cases and you should not overreact.
And if you look in corporations that compete with one another, one of the things that's missed in sort of corporate thinking is that corporations compete with each other, but they do in fact share common goals. And the fact of the matter is that employees move from one corporation to the other without being shot in the middle. Right? They're not imprisoned for moving from a company to another, even though they take inside their head the knowledge that they learned. And that's another bargain, right, that we get for free speech, free movement, free employment, which are obviously sacred. And so occasionally, yeah, you lose an employee to a competitor, and hopefully they won't steal information, and if they do, then you'll go after them. But the fact of the matter is the network of people that emerged as a result of that across the community is far stronger than if there had been hard boundaries where you couldn't exchange information. Lots of studies about the Bay Area have said that one of the fundamental reasons why it has moved in lockstep in the technology industry is because of that movement, right? That people could eventually move from firm to firm. It wasn't lifetime employment.
I'd like to give each of you the opportunity to ask each other a question. Eric, do you have a question for George?
What I'm interested in is what happened. What is the exceptional thing that will come out of your lab in 5 to 10 years, right? You should be able. And let me challenge you to say you should be able to answer this, because you've got graduate students now who in 5 years will become postdocs, and they will have done something extraordinary.
Right. So the cop-out answer is that we are so freewheeling and so out of the box that we surprise ourselves on a 5-year time frame. I think that even a few months before CRISPR, we would not have predicted where it went, or even a few months before next-gen sequencing. But I won't cop out entirely. I think that we will have probably breakthroughs in organs and antibodies and using machine learning for protein design. We already have that happening now, so I think 5 years from now, that's a safe prediction. I should ask a reciprocal question. I've noticed that a lot of let's say entrepreneurs and people who've done very well in the physics and computer science gravitate towards biology. Gates and Zuckerberg, now the Chan Zuckerberg initiative, and the list goes on. Elon Musk and his brain interface, and why is this, and what motivates you in that regard?
If you do this long enough, you get a sense of where the opportunities are, and you can't prove it, but you look at the rate of innovation. And I'm always interested in the following question: what has changed the fastest? What is the thing that is changing right now the fastest? Because whatever that is going to determine the history of next year, right? Because whatever that rate of change. And humans are very bad at estimating rate of change. And most things don't change, and a few things change enormously. There's a lot of evidence that biology is in that golden period right now, right? And again, you know the history better than I, but I think it starts with sequencing the genome, that kind of stuff. And earlier, of course, the original double helix discoveries and so forth way back when. It just seems like taking biology, which I've always viewed as squishy and analog, and turning it into something which can be digitally manipulated is an enormous accelerant. And I don't see the same. There's probably examples in chemistry where the same thing is occurring. I know there's examples in material science where that's occurring. But I think what you want to do if you're kind of scoring the rate of change thing is you want to look for things which have either a simulator that's digital, or they have enough data that is accumulated so that you can operate in the digital domain where the tools in my world can apply very quickly. And my guess is that's why people are so excited about it.
So I think their questions to each other are way more interesting than my questions.
Very modest.
So if it's okay, I would like to ask you to continue. Eric, do you have
So you talked about, okay. So you talked about organs. And people have talked for decades about the ability to grow human organs and so forth. And many people believe that that will occur in our lifetimes. So is it reasonable to think that in our lifetimes you'll be able to grow a human liver and do that as a liver transplant, or that kind of, the things which people are really bedeviled by these problems right now? How long will that take?
Well, so it depends on how much you cheat. So if you grow the organs in pigs, it's just a matter of humanizing the pigs to the point where they are as good as a matched, histocompatibility matched organ between two humans. We're probably pretty close on that. It has been a 20-year dream. It started long before I was interested in it. $1 billion was committed by Novartis and others, and it clearly required much better technology than we had 20 years ago. I think we're there now. We have them in preclinical non-human primate trials at MGH, so it could be at hand. We'll see.
So if it's sort of pre-human trials, then it's probably 1 to 2 decades away, given the nature of first trial, second trial, so forth.
Well, so yes, I mean a clinical trial. Even if we got 12 months, we have 9 months survival. If we had 12 months survival, then it might be a year or two before the clinical trials start. Then you're talking about typically 10 years before it's in wide use. Nevertheless, sometimes people get cured during the clinical trial, so it's a matter of definition. But yes, before widespread use is easily 10 to 20 years. It's hard to speed that up. Every time we try to speed that up, we end up with something like thalidomide or Vioxx or hormone replacement therapy. So we have to be patient. It is trial and error, and human lives are at stake.
George, do you have another question for Eric?
Yeah, so why did you ask 5 years from now? I mean, why not more or less? I mean,
Well, I'll say why 5 years, and it's my habit, it's my verbal pattern.
Yeah.
I find that people can't answer a question for 5 years. They can answer a question for 1 year, 18 months, and they can guess over 10 or 20 years. But almost all businesses fail because they don't ask the 5-year question. And in general, you have enough as a business person, you have enough in front of you to be able to accurately estimate what your 5 years look like. So in my world, you'll look at an example. Cost of semiconductors, cost of networking, cost of phones, right? Cost of software. You have a reasonable expectation of the cost of software and content and media. So you start putting, if you ask the 5-year question as a general rule for startups, you get a very good answer because it forces people to think well past their traditional planning horizon.
Yeah, I mean, in software you have, and hardware and computer industry, you have the ITRS roadmap, which tends to be, at a minimum, a self-fulfilling prophecy. In the Wild West of synthetic biology, we at least deceive ourselves into maybe lazy thinking that it's a little more unpredictable.
Well, let me answer that in 5 years, you will have figured out the digital platform that allows for the much faster loop that you have been discussing. I am certain with the quality of people you have and the number of people here in the room who are working on this, plus the fact that people are collaborating, there will be a quantum jump in acceleration as a result of this platform. That's why this is such a critical time in synthetic biology. How do I know it's 5 years? Because it takes a few years to build the platform, and then it takes a few years for everybody to pile on.
Right.
Well, fantastic. I'm sure that everybody else in the audience is as excited as I am to hear such a legend as Eric Schmidt get excited about speeding up the design, build, and test process with biology. Let's give a huge round of applause to both Eric Schmidt and George Church.
Thank you both.